Hello everyone, welcome on your YouTube channel of Lalbagh Girls Center College. This is Jyoti Singh. Class 12, in this part we are going to discuss about the DNA recombinant technology. So let's learn about the DNA recombinant technology, various terminology we are using in this video. So first of all, we see the definition of recombinant DNA technology, what are the major steps involved in this, what are the major techniques involved in this procedure. So let's learn about the DNA recombinant technology.
So biotechnology may be defined as the method by which a living organism or its parts are used to change or to incorporate a particular character to another living organism. Basically, it involves the applications of scientific principles to the processing of materials by biological agents. So we will see what are the biological agents, what are the techniques, different type of techniques are used in biotechnology. Genetic recombination is the exchange of genetic information. Basically, we can say that genetic recombination is the exchange of information between two DNA segments, and this is a common occurrence within the same species. But by artificial means, when a gene of one species is transferred to another living organism, it is called recombinant DNA technology. Basically, what we are doing is we are incorporating a different kind of or different set of gene into the desired DNA fragment. That means we are incorporating a desired segment or desired fragment of gene into the plasmid DNA. So it is a common method and this is known as a genetic engineering. Now we will see definition of recombinant DNA. So the production of a unique DNA molecule by joining together two or more DNA fragments not normally associated with each other. DNA fragments are usually derived from different biological sources. Now these DNA fragments we are derived from a different biological sources, and these biological sources are the living sources where we are cut off the specific sequence of the gene, and when we cut the specific set of gene, then we will incorporate into the new DNA sequence. Now see the definition of recombinant DNA technology. Last step, a series of procedures used to recombine DNA segments under certain conditions. A recombinant DNA molecule can enter a cell and replicate. That means when we incorporate the desired segment into the plasmid, then it goes for the recombination technique or cell get replicate. So after the replication of cell, we get the DNA fragment of that desired gene also. Now we will see the history of recombinant DNA technology. Recombinant DNA technology is one of the recent advances in biotechnology, which was developed by two scientists named Boyer and Cohen in 1973. Now we will see the development of molecular biology. Early research on prokaryotic genetics and development of molecular techniques has led to a new discipline that is called molecular biology, right? So the tools have been developed to enable scientists to examine very specific regions of the genome or gene. Now we will see advances in molecular biology. First is the combination of restriction or the modification enzymes and hybridization techniques enable the application of a wide variety of procedures. Now here are some kind of techniques and the procedures used in recombinant DNA technology. First is the gene isolation, purification and the synthesis. Next is the sequencing, genomics, or we can say proteomics. Next event in this technique is your polymerase chain reaction, which is also known as a PCR. Next is the mutagenesis, also called as a reverse genetics. After that, you will see the expression analysis, in which we will see the transcriptional and the translational level. That means we are analyze it at the level of transcriptional, at the level of translational level. So the restriction, next event we are seeing in this technique is the restriction fragment length polymorphism. Next is the biochemistry or the molecular modeling, and at the last we will see the gene therapy. So there are some common methods or common steps or the procedures in this technique. So we will see all these events, all these steps or the procedures or the technique we are using in biotechnology or DNA recombinant technology. So we will see its application also. We will see its application in the quantitative preparation of biomolecules, recombination vaccines, and the monoclonal antibodies, and the diagnosis of the genetic diseases, cell and tissue culture, and to identify mutations in genes, and after that we will see its application on the xenotransplantations. So children, this is all about the introduction about the DNA recombination technology.